Hello children, uh, hope you all are fine. You are doing well with all the online classes. I have come again with the continuation of the last chapter, number 2, that is reproduction in plants. Last time we did the artificial, we did asexual or vegetative propagation, its types, its advantages, disadvantages, all we did last time, and I had given you some assignments also. Perhaps you people did it. Now we'll do now the artificial vegetative propagation. All right, what will do it? Artificial vegetative propagation. All right. So by the term itself, you'll be knowing what we are going to do today. Okay, artificial means, without intervention, okay, without interference. Okay, we are going to propagate the plants. Okay, so when we are going to propagate the plants as per our want, as per our desire. So what kind of plants we are going to select? We are going to select those plants which are good, which are very much useful to us, okay, which brings about lot of benefit to us. So those kinds of plants are selected for the artificial vegetative propagation. Okay, so there we intervene. So last times what we did was all natural vegetative propagation, natural asexual propagation. Okay, so today we are doing it artificial vegetative propagation, where we interfere at our want. Okay, at our want, the plants are being propagated. So what kind of plants? Plants those are useful to us. All right. So this artificial vegetative propagation have been grouped in 4 ways. That means artificial vegetative propagation can be done in 4 ways according to the points given in your book. There are many, but here we do the 4 types of artificial vegetative propagation. All right. So number 1 is cutting. Number 1 is cutting. Number 1 artificial vegetative propagation is cutting. So in cutting, what we are going to do, let's see. All right. Auxiliary bulb, some node. Okay. So cutting, how we can do the cutting is that we select the plants. Okay, for example, rose. We have rose. All right, we have rose, sugarcane. All right, then China rose. All right, then we have lemon, etc. Okay, so we select those plants. We said there are other plants also where cutting can be done. Okay, so let us take, for example, these plants as I have written out here: rose, sugarcane, China rose, lemon, etc. So these plants are selected, and from there, a small stem is cut. Okay, as I have shown out here, the small stem is cut with some auxiliary buds. Some auxiliary buds must be there with 1 or 2 nodes. Node is there, at least with some auxiliary buds and node. We cut it, we make the cutting of it, then we plant it in a moist soil. We plant it in a moist soil or fertile soil. All right. Then what we do is that we frequently we can water the bed where we are doing this cutting also. Then after some weeks, after 4 or 5 weeks, what happens is that from here the portion which is buried in the soil, so there inside the roots are developed. Okay, the roots are developed. Then the plants, this cutting will be ready to get transplanted. So how we can come to know about the success of this cutting is that when the rootings are taking place out here, okay, when the rooting is taking place out here, this axillary bud, the new leaves will all be formed, okay, they will all be growing. So once everything is grown well out here, this axillary bud is seen well developed, node is well developed. When it is seen, then we'll be making sure that the cutting is now successful, okay. Then when it is successful, then we can transplant it to the main field. Then we can transplant it to the main field. Okay, so this is called cutting. Alright, so which we can do with the plants like rose, sugarcane, China rose, lemon. So in those plants we can do the cutting. Alright, then we have second is layering. Layering. So second is your layering. Alright, so layering. Okay, so it's better to draw the diagram and explain this so that you people understand it well. All right. Right. So okay, layering. So this is a plant which is already fixed to the soil. So in layering, what you can do is that we'll select the branch which is at the bottom, the lowermost branch will be selected. Okay, then what to do is that there in certain region, what you can do is that we can remove the bark. Okay, we can remove the bark. Bark can be removed with a sharp knife. Then we pull it down. Okay, we pull it down like that. We pull it down like that. Then we bury it with the soil like that. Okay, now this is there. Okay, so we cover it with the soil now. Here, this portion is covered with the soil. All right, this portion is covered with the soil. Okay, so to fix it there, we can even put some heavy object like brick or some other little heavy object can be kept and press it so that it doesn't shake. Okay, so over the course of time, what happens is that here the portion which is buried will develop the roots out here. Will develop the roots. All right. So when the root is developed, when you are sure that the rooting has already taken place. Okay, looking at the condition of this region, when you are sure, then what you do is that you can cut it up. Okay, you can cut it up and you can plant it here. Okay, you can plant it here. So this is called layering. All right, this is called layering, which can be done in the plants like rose. Again, even in the rose, this layering can be done. Even in the plant like mint, all right, jasmine. So in those plants, this layering can be done. Then third type of artificial vegetative propagation we have is, all right, we have grafting. All right, grafting. All right, grafting. So in grafting, what we can do is that, especially in grafting, we usually select two different varieties of the same plant. All right. Sometimes what we can also do is that grafting can be done sometimes in between the two different closely related species of the plants also. Okay. Suppose you see the lemon and orange plant. Okay, these two are the two different species of the plants, but grafting is possible there. Okay, so especially in those plants where the genetic compatibility is there, so there the grafting can be done. Okay, so that whatever we produce from grafting can be a new type of plant, can be a new variety of the plant, can be a new type of plant. All right, where we can obtain the properties of both types of plants. Okay, so that benefit we can have in grafting. So how we can do grafting? Okay, let me tell you with the help of this diagram. Okay, so for grafting, I told you we need always two varieties of the plant. Two varieties of the plant of the same type, or actually even the two different plants of the closely related species. So there also it is possible. Okay, so for which we need to have two portions of the plant. First we have this one. One plant out there. So this is a plant which is already fixed to the soil. Okay, then so this is growing there. Let's see, this is growing there. Okay, one is selected, and for the grafting we can cut it here and we can remove this one. We can just remove it. We'll remove it. Okay, so we can just remove it. We just cross it. All right, then we can, after removing that, we can cut this middle portion of this plant in this form. So this middle portion of this one can be removed. Okay, it can be removed. All right, it can be removed. So like that. So this portion of the grafting which is fixed to the soil is called stalk. All right, the stalk. So if there is any leaf or this one can be removed, we just remove them. All right, we just need this stem outer which is fixed to the soil. Then on the other side, the other variety of the plant is selected. Okay, other variety of the plant is selected. So they are growing here. They are growing here. So like that. So they are growing here. Then what you can do out here is that this person we cut it. We cut it here. We cut it. We don't need it. We just take this one only here. We just take this. So taking this one, in what portion it is cut? So we need to cut this portion also in such a way that it can be fitted here. It can be, it can be nicely articulated out here. So that we take out here, then we cut it in this position. We cut it in this position. So these plants are there already. So this would be taken. This is called scion. S-C-I-O-N, scion. So this now will be taken here. Okay, it will be articulated here. Articulated here. So like that. Okay. After fixing this scion into it, okay, what we can do is that we can bind it with the help of tape. We can bind it with the help of tape. Okay, we'll bind it very tightly with the help of tape so that they are not shaken, they do not move. And also what we can do is that we can apply some wax out here. We can apply some wax here. Okay, in order to check the dehydration and also microbial infection, what we can do is that we can apply the wax so that dehydration can be checked and also microbial infection can be checked out here. All right, and when you go to articulate this scion into this stalk, you make sure that they are very nicely articulated, so that there is a very good translocation of the things from here to this and from here to this, that is vice versa, because this region which is called stalk has to supply the water minerals to this upper region. Now after using the water and minerals, whatever they prepare out like food and all should be supplied to this also, so that very good, very smooth translocation of the materials should be taking place between these two, stalk and xylem. Okay, so over the course of time, the very good development will be taking place out here, and this graft will be growing as a single individual plant. Okay, doing this, what we can get is that, what benefit we reap from this is that here the characteristics of these two plants can be obtained in the single plant, or in the new plant we can have the properties of both these varieties of the plants. Okay, so it may be like this. Suppose this which I have taken as stock, it may be very much good in terms of in terms of fighting with the diseases. They may be very good drought resistant, they may be very good disease resistant. Okay, and the other one which I have chosen as sign, that may be very good in giving you the production. It may be bearing very good fruits in large size, sweet and very juicy. So that quality it may be having. So these, both the properties from xylem and stalk can be obtained in the single plant. So that is the very good benefit that you can reap from grafting. So I have told you this is possible in many of the plants, like even rose, it is possible there. Even in mango it is possible. Even in guava it is possible. There may be guavas of different varieties, but all those varieties of this guava can be grafted. Okay, even I have told you that the different species of the plants, that means closely related species of the plants, can be taken for the grafting. Okay, that is very good for us. Then we have got number 4. Then we have number 4. We have number 4, micropropagation. Micropropagation. Okay, micropropagation is a tissue culture. In fact, is a tissue culture. Okay, tissue culture. All right, tissue culture. Micropropagation is a tissue culture only, where it is applied to those plants where the vegetative parts cannot be used for the propagation. Okay, so in such cases, this micropropagation can be done, like in the case of gladiolus. It can be done in gladiolus. All right, orchid. In such plants, this micropropagation is done, where the big vegetative part is not needed. Some cells from the shoot apex, some cells are taken, some tissues are taken, and that can be done in the small area also. It's not that you need big plot of land for doing this micropropagation, but in the small place only this micropropagation is possible. So how we can do it? Okay, let me tell you with the help of these diagrams. I'm going to draw the diagram first, then I'll let you know how we go about. Okay, let me draw the diagram first. Okay, so this is the flax web. Okay, this is the nutrient medium. All right, then here the some explants. Okay, like a shoot apex. Shoot apex is explant. Explant is taken. Explant. All right, and cover it. This one is cotton. All right. Then second stage. And the second stage. nutrient medium is there, same nutrient medium. Alright, then the callus formation. Callus formation. Okay, that is there now. Callus formation. Then third stage. I'm going to do third stage out here. The third stage. Embryo is formed, still cotton, then this is plantlet. Okay, this is this is plantlets. Okay, plantlets after 4 to 6 weeks. Okay, so now this is how this micropropagation is done. First, as I've already told you that you don't require a big plot of land. So this is usually done in the tissue culture center, like in the research center, as you have there in nearby this parking airport. Also there is ICR, okay, orchid house there, where usually tissue culture is done. Okay, so even there this is done. All right, so for which what you can do is that you can take one small what do you call it, you can take it one plaques as shown there in the diagram. Then there inside the plaques there you have prepared a nutrient medium. Please remember nutrient medium. So nutrient medium may be something like solid, but that should be rich in nutrient. Okay, so nutrients required by the living cells out here. Okay, living cells. Okay. So here, before preparing all those things there, what you have to see is that you need to get the shoot apex of the plant of which you are going to do the micropropagation. Suppose you are going to have the micropropagation done of these, gladiolus or orchid, okay, or you are going to do the micropropagation of orchid. So in that case, what you do is that you take the root apex of gladiolus or orchid. And that part which is called root apex. Root apex means tip of, shoot apex. Shoot apex. As I've written, shoot apex. That is tip of the shoot is taken. Okay, so that is called explant. Okay, technically that shoot apex which is taken for this purpose is called explant. E-X-P-L-A-N-T, explant. This explant, in this case we have taken shoot apex. So shoot apex is first treated, okay, is first treated with the sterilizing chemicals, because this shoot apex may be, infected with some kind of fungal disease. There may be some bacterial problems. Okay, so in order to disinfect this, in order to, make it free of such microbes, this shoot apex is first treated with such sterilizing chemicals. Okay, then after that, from the shoot apex, some cells or tissues are taken out. Okay, and that cells and tissues are kept on this, or are cultured on this nutrient medium. As I was saying about nutrient medium is the place where we culture this, shoot apex, or the cells taken from the shoot apex, and that nutrient medium is very rich in nutrients required for these cells, because the cells which we are using out here are all living. All right. then at the expense of the nutrient from here, these cells now grow up. They may be, very rapidly undergoing the cell division, and after cell division, they may be, differentiating, they may be growing, all right? Then what happens is that there will be a cluster of cells formed, okay? A cluster, dense cluster of cells will be formed in the next stage, which is called callus, C-A-L-L-U-S, callus, okay? So callus is formed here now, all right? Then after the callus formation, okay, so further the cell differentiation will be going on, and it will be further growing, and in the third stage, embryoid is formed, embryoid, okay, embryoid. Then from the embryoid in the last stage, stage, what is formed is that the small, several tiny plants are formed. From the cells, you see, the cells were used out, some tissues were cells, or cells were used, or tissues were used out here, will be ultimately developed into the tiny several plants, okay? Several tiny plants are developed in this stage, okay? And these several tiny plants which are formed out here are called plantlets. Now see, plantlets, okay? Are called plantlets. So after 4 to 6 weeks, okay? After 4 to 6 weeks, these plantlets are taken out and grown in the spots, or taken to the place where we grow them permanently. After 4 to 6 weeks, these plantlets are taken out and grown in the pots, or taken to the place where we grow them permanently. Okay, so this is how the micropropagation is done. So these are the 4 different types of artificial vegetative propagation, and this is then these 4 different types of artificial vegetative propagation that we can do. Now we'll go through the benefits, why we go for this artificial vegetative propagation, what are the significances. Okay, let us talk about benefits first. Number 1, so rapid propagation of identical individuals. Okay, rapid propagation of identical individuals. So, by this method, by the artificial vegetative propagation, we can rapidly produce the similar kinds of plants. Similar kinds of new plants can be produced very rapidly. So as I've already told you that what kind of plants we are going to culture out here, what kind of plants we are going to propagate out here, those which we want. So all those good identical plants can be rapidly produced, number 1. All right, and number 2, inter-specific hybrid is possible. Hybridization is possible. All right, what is that? Inter-specific hybrid is possible. Inter-specific, that means species. You just got to know what a species is. Species. So species, I have already told you that in the class also. Species, okay, so when the different members of the organism where interbreeding is possible, that is a species. Okay, like human being is a species. Okay, mango is a species. Okay, guava is a species. So in this case, in the artificial vegetative propagation, though not naturally, I'm not talking about naturally, okay, artificially we can even hybridize the 2 different species of the plants. But what kind of species of the plants? Plants which are closely related species. Okay, maybe the different species of the plants from the same family. Okay, like in the case of, here I've told you before, the orange plant and lemon plant. Okay, they can be hybridized by the grafting. So that is possible. Okay, that is possible. That means in those plants, in those different species of the plants where genetic compatibility is there, so there this your artificial vegetative propagation can be done. Okay, that is only artificially, but not naturally. Like in the case of animals also, we can hybridize between the male donkey and female horse to produce a new kind of animal, new kind of offspring, that is mule, M-U-L-E, mule. Mule is an animal produced from the cross of male donkey and female horse. So that is possible there, not naturally. Artificially that is possible. In the same way, here in the case of this also, we can produce the interspecific hybrid. Okay, interspecific hybrid can be produced by this artificial vegetative propagation. Alright, number 3, it is very useful in those where seeds are dormant. Okay, where seeds are useful in those where seeds are dormant. That means there are many plants, though they have seeds, though they have fruits, where the sexual reproduction is not possible. They cannot reproduce on their own. Okay, so in such, if they do also, it takes very long time and the success rate will be very less. So in those cases, what we can do is that we can go for artificial vegetative propagation. Okay, so artificial vegetative propagation can be applied to those plants where the seeds are dormant. Okay, where the seeds are not that active. Alright, so these are the benefits. Okay, then though these are the benefits, there in the same time there are some limitations. Okay, there are some limitations of this artificial vegetative propagation. Number 1, they are not applicable to all cases. 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 Alright, not applicable to all cases. As I have told you that grafting, where we can cross two closely related species of the plants. Alright, but it's not that you can cross all species, different species of the plants, you cannot do. Okay, you need to know that where the genetic compatibility is there. Okay, in which different species of the plants, the genetic compatibility is there, which are closely related. You need to know that. Then only that is possible. It's not that having the idea of that if you are going to cross one type of plant with the other species of the plant, if you are going to do that, maybe not successful. So in order to check that, you need to know that where it is possible. If they are closely related, then only it is possible. So not applicable to all cases. Second, what is there is that not easily applicable in remote areas. Not easily applicable in remote areas. Okay, so for example, that micropropagation is not possible everywhere. You need so many things out there. You may be needing the plant growth, or plant hormone and all that we have used there in the micropropagation, that you require in the micropropagation. Sterilization chemicals, the different kinds of equipments we may be needing. So all those things may not be available in the remote areas. Okay, so that means this is all not, some of these may be useful, but all different forms of that artificial vegetative propagation may not be possible in the remote areas. So this is your limitations. Okay, so that's all about your artificial vegetative propagation. I hope you people have understood it, and once again I would like to request you all, please go through it, not only once, not only twice, not only thrice, as many times as possible. Okay, to make it very clear, you just go through it as many times as possible, and once you go through it, you please keep your book open so that you understand it. Okay, I do hope that you people do the same, that's what I am telling you. Alright, so now I just want to give you some home assignments. I hope that you people do it. So last time also I given, you must have done it. Okay, home assignments number 1. Write short notes, own, cutting, alright, then we have layering, alright, grafting, and micro-propagation. Right? Then number 2, define. Define. What you do is that explant, callus, stalk, and scion. Okay, so these are the homeworks for you. Number 1, write short notes on cutting, layering, grafting, micro-propagation. Here diagram is important. Diagram is a must. Okay, we need to draw the diagrams of all those here. That's very important. Then define, explant, callus, stop, and sign. Alright? So these are the homeworks for you.